In this video, we will see an activity to show that CO2 is necessary for photosynthesis. We know that plants obtain CO2 from the atmosphere through openings known as stomata and perform photosynthesis. How do we demonstrate it? First, we have to take destarched potted plant, that is, we have to keep it in darkness for about 48 hours and remove all the starch. We have to take a conical flask and put some potassium hydroxide into it. Now we know potassium hydroxide absorbs CO2. Now insert one leaf into this conical flask through a split cork as shown in the figure. So except this leaf, all the other leaves will be outside of the conical flask. Leave the setup in sunlight for few hours. After few hours, we have to take out this leaf which is present inside the conical flask, and we have to take one leaf which is outside the conical flask. So they both will be tested for presence of starch. Now the observation is that the leaf kept in the conical flask does not turn bluish black when we put the iodine solution, whereas the leaf which is present outside, this will turn blue black, and proves that only the leaf which is outside or which is not exposed to potassium hydroxide will turn bluish black. Now inference is this shows that CO2 is necessary for photosynthesis. Now we will see another activity to prove that CO2 is necessary for photosynthesis, and this experiment is known as Moll's half-leaf experiment. Now we have to take a leaf from a destarched plant which was kept in darkness for 48 hours. Insert only half of the leaf into a wide mouth bottle containing KOH, and this KOH solution in the bottle absorbs CO2 from the air present in the bottle. So half of the leaf will not have access to CO2, whereas other half will have access to CO2. Now we have to dip this cut end of the petiole into a petri dish containing water, because for photosynthesis water is necessary. Keep the setup for 4 to 6 hours in sunlight. Then we have to take out the leaf and perform iodine test for presence of starch with this leaf. Now the observation is half of the leaf which was out of the bottle, that is this part which is not dipped in the KOH, will turn blue black with iodine solution. Now the other half which was inserted into the bottle does not show any change in color with iodine solution. So we can tell that the part of the leaf which was dipped in KOH is negative for starch test. Part which was not dipped in KOH is positive for starch test. The inference is this shows that CO2 is necessary for photosynthesis. I hope this topic is clear to you. Thank you.